Deoxyribonucleic acid, or DNA, is the biological blueprint of a human being. It relies on a simple four-nucleotide system: adenine, thymine, cytosine, and guanine, and it resides in almost every cell of your body. This table compares standard identification modalities. While fingerprints or iris scans balance moderate speed with acceptable error rates, DNA sits at the extreme ends. It yields the highest possible accuracy, but at a very high monetary cost and incredibly slow processing speed. This creates a mechanical paradox. We possess an identifier that is physically exact, yet completely unsuitable for real-time authentication. You cannot use it to unlock a phone or clear an airport security checkpoint. The delay occurs because DNA identification is a rigorous, multi-step signal processing system. To read a genetic signature, forensic engineers must take messy, physical biological material and chemically force it into a discrete digital template. This diagram maps the required architecture. The pipeline moves from sample collection through extraction and amplification, detects variations, generates a profile, and executes a match. We must dismantle this engineering pipeline step by step, starting directly with the physical evidence. The system begins by gathering physical biological evidence, typically blood, saliva, or hair roots. At this stage, isolating the sample and avoiding external contamination is the strict governing rule. Engineers immediately face a data processing problem. A complete human genome contains 23 pairs of chromosomes. Analyzing all of this genetic information to confirm identity is highly inefficient. Instead, the biometric system targets a highly specific genetic variation known as short tandem repeats, or STRs. This diagram illustrates an STR, sequences of base pairs repeating consecutively at locations called loci. Most human DNA is identical, but repeat counts vary radically between individuals. At each locus, a person inherits two repeat counts, one from each parent, known as alleles. Isolating these specific STRs from a collected sample requires precise physical extraction. The mechanics begin with cell lysis, physically breaking the cell membrane. Technicians then utilize protease enzymes or silica-based methods to strip away the unwanted cellular proteins. The output of this stage is pure, isolated genetic material, cleanly separated from cellular contaminants based on specific solubility or ion-binding differences. This chemical extraction acts as an analog filter. It strips away biological noise, leaving a pure DNA sample ready for exact measurement. Before processing the pure DNA, the exact concentration of the extracted sample must be verified through a step called quantification. This chart plots the principles of spectrophotometry. Pure DNA absorbs UV light, specifically at a 260 nanometer wavelength. By measuring this absorbance, technicians calculate concentration. An absorbance value of 1 equals 50 micrograms per milliliter. Verifying this concentration standardizes input and ensures statistical reliability. With the concentration verified, the system must solve the problem of microscopic sample sizes using polymerase chain reaction, or PCR. PCR amplification targets the specific STR regions we need to measure and forces them to produce millions of identical copies. This maps the PCR cycle's first step: denaturation. Heating to 95 degrees Celsius unzips the double helix. Second, in annealing, temperature drops, allowing markers to bind to targeted STRs. Finally, extension heating duplicates the marked segment. Repeating this cycle yields an exponential explosion of STRs. PCR acts as a biological amplifier. Overcoming the physical limitations of tiny forensic samples, it generates enough raw material for digital sensors to read. The engineers now face a logistical hurdle. They have millions of microscopic amplified fragments, and they must count the exact number of sequence repeats within them. To bridge the gap between biology and data, the pipeline relies on capillary electrophoresis. The amplified DNA fragments are tagged with fluorescent dyes. As they move through the system, a detector reads the fluorescence to measure their exact physical length. Because the sequence lengths are strictly uniform, measuring a fragment's overall physical length allows the system to definitively calculate the exact number of STR repeats at each target locus. At this exact moment, the physical molecules are discarded. The biological material is fully converted into a digital numeric template. This table represents the digital biometric template, plotting specific loci against inherited alleles. At locus D8S1179, this individual has one sequence repeating 12 times, and its counterpart 14 times. This structured matrix guarantees privacy. It contains no health information, just a compact numeric sequence. To build a legally binding profile, forensic databases typically track between 13 and 20 of these specific loci. The messy biological reality of a human cell has been translated into computationally efficient code, primed for mathematical analysis. With the digital template generated, the focus shifts to the matching algorithm used to compare the profile against massive forensic databases. The foundational rule of identity matching is absolute. Confirming a subject requires exact numeric allele matching across all tested loci. This leads directly to the critical statistical question that gives DNA its legal weight. If a match occurs, what is the exact probability that it happened purely by chance? This formula calculates random match probability, or RMP. Because each genetic locus is inherited independently, their probabilities are multiplied together. A 1 in 10 chance at a single locus isn't unique, but multiplying that chance continuously across 15 loci causes the statistical denominator to rapidly explode. The ultimate statistical threshold is often an RMP ratio of 1 in a trillion or lower. Reaching this number effectively rules out a chance match, providing probabilistically unassailable proof of identity. This exposes the overarching trade-off of the entire system. Achieving this level of absolute mathematical certainty requires intense chemical extraction, thermal cycling, and complex computational analysis, demanding thousands of dollars and hours of laboratory processing. While optical biometrics optimize for immediate convenience, DNA biometrics sacrifices speed entirely to guarantee the absolute, mathematically unforgeable truth of human identity.